Hello children. Plants are struck at one place. Still, they have an ability to reproduce sexually. This is something which is very surprising, isn't it? We'll be discussing at length about these aspects in a chapter called Sexual Reproduction in Flowering Plants. At last, we enter into the second year, and we'll try to complete this very fast. Now, the first part of it is, when can a vegetative shoot transform into a reproductive shoot? Listen carefully. Leaf is a place where a flowering hormone is synthesized. From the leaf, this hormone migrates into the shoot apices, and now the shoot apices transform into a floral primordia, which later develops either into a flower or into an inflorescence. So it means that the part of the plant which is very essential for flower is leaves, because hormone responsible for flowering are actually synthesized in leaf in certain photoperiod, and gibberellins also have a role in the synthesis of flowering hormone. So that's what I have written here. Leaves perceive light, all right, and help in the synthesis of flowering hormone. Remember, florigen. This hormone is transferred to the shoot apices, transform them into a floral primordia. That's a very easy one to remember. We made a good start, isn't it? Now, where are the flowers seen? Some easy points will run very, very fast. Observed in angiosperms and in gymnosperms, you call them as cones, strobilus. The reason why I call it cone, because it doesn't have any perianth, so therefore I should not call it as flower. Morphological marvel and embryological, truly. Now, it is seen in all sexually reproducing flowering plants. Saying that it is seen in all sexually reproducing organisms is wrong. Alga, bryophytes also reproduce sexually. Sexually reproducing flowering plants. Nature, it is modified condensed shoot. Why I say shoot? We already discussed in flower chapter. This is the stem. The stem should have nodes, 4 nodes. The first node contains sepal, second node petal, third node stamen, and fourth node carpel. This long stem becomes condensed. Then all the floral parts appear to arise from the same point, as you could see here. Then this is said to be a flower. Why I call it a shoot? Because it contains stem and leaves. What is the highly condensed stem called? Thalamus. And these floral whorls, all the sepals put together called calyx. Each member is sepal. All the petals put together is forming a whorl called corolla. All the stamens put together forms androecium, and all the carpels put together forms gynoecium. So floral whorls, these floral leaves are these. Well, you know this. This is the typical flower, children. Receptacle is a term used for thalamus. All right, this is the stalk of the flower called pedicel. The thalamus may be conical, the thalamus may be saucer shaped, it may be cup shaped. We studied in the flower chapter. Now it is time for us to talk about next important unit. What a flower contains. A flower contains stamens. Question you're going to ask me is, why stamen is said to be microsporophyll? Why you call them as sporophyll? Phyll means leaf. Anything which arises from node is a leaf. Stamen is arising from the third node. Therefore, I want to call it by one name, sporophyll, microsporophyll. Isn't it? Yeah. Then, what is the stalk of the stamen called? Filament. What is the swollen spore-producing region of the stamen? This is the swollen spore-producing region. This is the filament, and this is said to be anther. The proximal end, this is the proximal end. The proximal end of the filament is joined to thalamus or petals, if it is epipetalis. All right, or the distal end is joined to fertile anther. This is the proximal end, joined to thalamus or with petals. Distal end is joined to anther. Very important point this is. Then a dithecous anther possess. This is an important point you have to remember. Look here. I take this as a leaf, okay? Leaf. I tear it into 2 parts. Each half I am folding like this, okay? Look here, each half I am folding like this. So this is one half which I folded. Look here, children. This is the lobe. Careful, lobe. One part, one half. And you know, one more half was also there. So wish I don't have. I have one more book I don't have. So therefore, one more lobe. This is one more lobe. So one lobe on this side, one lobe on this side. So now tell me, how many lobes are present here? One on this side, one on this side. There are two lobes. Two lobes, two anther lobes. This entire thing is anther. And there are two lobes. You can easily remember this. Now the next important one. How many chambers or theca are present? In each lobe, 1, 2. On this lobe, 1, 2. So total, there are 4 theca. Theca means chambers, 4 theca. 2 anther lobes and 4 theca. This theca later becomes microsporangia. Later they become pollen sacs once the pollen grains are produced. So a typical anther, now I'm going to show you. A typical dithecous anther produce 2 lobes. Yes, this is one lobe. This is the connective. And this is another lobe. So therefore, it has 2 lobes. It has 4 theca. 1, 2, 3, 4. 4 theca. 4 pollen chambers. 4 pollen sacs. And it contains many microspore mother cells inside this sporogenous tissue, give rise to many microspore mother cells. Then they undergo reduction division and give rise to many microspores. So this is a typical anther, children. This is a typical anther which is easy to follow. The word dithecous is actually confusing, giving an impression that the entire anther has 2 theca. Be careful. Entire anther have 4 theca. Then why dithecous? Each lobe has 2 theca. That is the reason why it is called dithecous. Can you guess what is a family with monothecous anthers? Yes, children, you told correctly, though I can't hear. It is Malvaceae. The family Malvaceae, hibiscus family, shows monothecous reniform, kidney-shaped reniform anthers. Now that's about covered by 4 wall layers. We'll be discussing it. Many microspore. These are the points which you have to remember with regard to stamens, children. Now I'm going to show you how the stamens develop into what is called microspore mother cells. The longitudinal groove runs lengthwise, separating the 2 theca of anther lobe, isn't it? This is a longitudinal groove. This is one lobe. This is a longitudinal groove which is going to break like this to liberate the pollen grains. This is one more lobe, no? It is going to break longitudinally like this to liberate the pollen grains. So this is said to be a longitudinal groove which extends all along the length of one lobe, one more longitudinal groove all along the length. This is easy to remember. All right, now let me explain you how the stamens are going to produce the pollen grains and sperms. Ultimately, the stamen is a male sex organ. Let us see how it produces a pollen grains. So remember, children, in Plant Kingdom chapter, I told you a small table. I said sporophyte, and I told you the job of sporophyte is to produce sporophyll. Yes, sporophyll means leaf. The job of sporophyll is to produce bags called sporangium. And the bags, the job of these bags, inside these bags, there are spore mother cells. These spore mother cells undergo reduction division to give rise to spores. We'll take a small break here. Sporophyte produces sporophyll. Sporophyll produces sporangium. Sporangium produces spore mother cells. Reduction division to produce spores. In plant kingdom, do you remember in bryophytes, foot setae capsule is sporophyte. Capsule is comparable to sporophyll because it may turn green just like a leaf. Inside the capsule, there is a bag called sporangium. Inside that, there are spore mother cells. They undergo reduction division to give rise to spores. So easy to remember. Go back to ferns, pteridophytes. Adventitious roots, rhizome, and pinnately compound leaf is a sporophyte. Pinnately compound leaves called fronds are sporophylls. On the leaves, on the lower side of the leaves, there are brown colored dots called sporangia. They are bags which contain spore mother cells, reduction division to give rise to spores. Spores get liberated, they develop into a heart-shaped gametophyte called prothallus. They have antheridia, archegonia, fused to form zygote, embryo, and again sporophyte. Easy to remember. So that is a life cycle which have become ridiculously simple after going through this table. Now we put this table even for angiosperms. Let us see, all right, whether we can do it for angiosperms also. The first one what we have is sporophyte. Sporophyte. Now what does the sporophyte contain? Root, stem, leaves, and flowers. Root, stem, leaves, and flowers. Root, stem, leaves, and flowers. That is sporophyte. This is the sporophyte. So sporophyte is over. We have completed sporophyte, children. We have done with sporophyte. Root, stem, leaves, and flower is sporophyte. Then what the next one we have? Sporophyll. I already told you, the stamen is said to be sporophyll. Why I told you it's sporophyll? Because it arrives from the third node of the thalamus. Anything which arises from node is a leaf, sporophyll. Now what do you see after sporophyll? Sporangium. If you take a transverse section of it, this is one lobe, this is another lobe, this is the connective which joins the two lobes, children. Yes. Now these are the four theca which later become microsporangium. You know what is the meaning of GIUM? It stands for bag. Bag. These are the bags which are present inside that. Therefore, this is called micro. So we have completed sporophyll, we have completed sporangium. I am using a prefix microsporangium. In place of sporophyll, microsporophyll. That is the only difference. Then you see spore mother cells. Inside that, there are many spore mother cells. Many. All right. They undergo reduction division. Spore mother cells undergo reduction division and the spores are produced. And this is a spore which is produced, which is said to be the microspore. And these are the spores which are produced. And these are the spores which get liberated. Sporophyte, sporophyll, sporangium, spore mother cell, and spore. So easy to remember. It is turning out to be easy. Now, what will happen to this spore before it is getting liberated? What will happen? You know, this spore is called pollen grain. And all of you know, the pollen grain is covered by two wall layers. Outer wall is called exine, inner wall is called intine. At some places, exine wall, sporopollenin is not deposited. Those areas, you call them as germ pores. Inner intine wall is made up of cellulose and pectin. 25 to 50 micrometers. Pollen grain I'm talking about. It has dense cytoplasm. It has got a large vacuole. What will happen to this microspore? First, we should go back to the table, children. What spore should give rise to? Listen carefully. The job of spore is to give rise to gametophyte. And the job of gametophyte is to produce gamete. So we have just up to here we have completed. Up to here we have completed. Now the spore should give rise to gametophyte and gametes. That's all. We have to get rid of these two. Remember this. Now what will happen to this spore? It should give rise to gametophyte. What will happen? Look here. Because of large vacuole, the spindle apparatus is pushed on one side. You see the formation of two cells. This is the large irregular nucleus. Irregular nucleus on this cell. And this is a microspore with two cells. In more than 60% of the families, pollen grains get liberated at this two-cell stage. From where? From the anther. What will happen to the anther, children? Look here. This is the anther wall. All of you know that. This is the wall, anther wall. What happens to this anther wall? There is one layer called endothecium. That will lose water. When it loses water, then what happens? It is going to become light and it is going to break. In the presence of high temperature, the cells lose water. When the cells lose water, the cells bend inwards. Look here, they are hygroscopic, they bend inwards. When the cells bend inwards, along the longitudinal groove, the pollen grains get liberated. This is on one side. For one more lobe, the endothelium cells lose water. They become light and they bend inwards. When it bends inwards, the pollen grains get liberated. And at this 2-cell stage in 60%, 3-cell stage in 40%. After it getting liberated, where it's going to reach? The stigma. What is this process called? Pollination. After reaching the stigma, this vegetative cell from the germ pore produce a pollen tube. The vegetative nucleus migrate, later degenerate. The generative cell migrate into the pollen tube and undergo mitotic division and produce 2 male gametes. Finished. Let us look all the stable children. First is sporophyte, yes. Then sporophyll, yes. Then sporangium, yes. Then spore mother cells inside, yes. Then spore or pollen grain, yes. 2-celled pollen grain, partly developed male gametophyte, yes. At this stage, it's liberated. Then this is the large vegetative cell, mind you, this is the large vegetative cell producing pollen tube. 1, 2, 3. This 3-celled pollen grain is said to be fully developed male gametophyte. Then you see male gametes. This is so easy to remember. Sporophyte, microsporophyll, microsporangium, microspore mother cell, microspore, partly developed male gametophyte, fully developed male gametophyte, and male gametes. Very easy. Nothing is there in this. Let us now look all the points. Now you can easily, we'll run through this topic now. The longitudinal groove run lengthwise, separating the theca of one anther lobe. It's a place of dehiscence. Hygroscopic nature of endothelium will help. 2 anther lobes are joined at connective, which we have seen this. This is the connective, this is the connective, this is the longitudinal groove. All right, this is easy to remember. Anther is tetragonal. Look here, this is tetragonal. All right, whereas each microsporangium is more or less spherical. This point is important. Microsporangia develop into pollen sacs. Initially, it contains sporogenous tissue. They give rise to microspore mother cells. They undergo reduction division and give rise to microspores. All right, did I say microspore mother cell? Yeah, we discussed microspore mother cell and produce microspores. Yeah, then microsporangium develop into pollen sacs. Then each cell of sporogenous tissue is a potential sporogenous tissue, potential pollen mother cell or microspore mother cell. Look here, this is microspore mother cell. It undergo reduction division. All the 4 microspores are covered by a common callose wall. This is said to be microspore tetrad. So if they ask you how many tetrads develop from microspore mother cell, your answer should be 1. Then all the common wall dissolve and the 4 microspores get liberated. This is easy. This is young anther. You could see the 4 theca. This is a mature anther. Why I say mature anther? By the time tapetum, the innermost layer, and middle layers degenerate, you could see children, you could see why it is mature anther. You could see this is breaking along the longitudinal groove, transverse section, breaking along the longitudinal groove for the 2-cell pollen grains to get liberated. Now let me talk about the pollen grains. Remember, pollen grain means microspore. This is important. Their yellow in color is important. It is the first cell of male gametophyte, yes. Pollen of different species could differ. This is not there. Pollen of different species could differ in shape, size and design, even color, but not in ploidy. All the pollen is haploid. They may differ in shape, size and design, and even color, mostly yellow. Exine wall made up of sporopollenin, important. Fossilization of pollen, important, is because of this. Pollens can stay as fossil for long because of this. Inner intine is made up of pectin and cellulose. Inner intine is made up of pectin and cellulose, and it produces pollen tube. We already discussed. This is said to be siphonogamous type of oogamy. You could see, children, these are the germ ports at which the exine wall is not deposited. You could see the generative cell smaller, vegetative cell larger, irregular nucleus. This is simple to remember. And then pollen grain. It has got dense cytoplasm with large vacuole, and some important examples. By this time, you are very much know all these points. Parthenium. Whenever you move on the countryside, when you pull your the windows down, you sometimes start sneezing because the pollen grains will be there in the air. Parthenium. All right, white colored flowers. And it's a Asteraceae member. All right. And this is called carrot grass. I forgot to write the name, common name. And it came to India along with imported wheat. We did not bring this, okay, accidental entry. And long viable pollen. Pollen is viable for many months in Fabaceae, Solanaceae, and Rosaceae. In your syllabus, there are 3 families: Fabaceae, Solanaceae, Liliaceae. Remove lily and put rose. Grass family means Poaceae. Pollen is viable for very less time. Pinus, winged pollen. Look here, the outer exine extends out like this to form a wing-like structure. Don't think that just like birds, it has wings. All right. And sticky pollen and edible pollen is seen in insect pollinated. All right, sticky pollen and edible pollen. And mind you, even the pollen is rich in nutrients. Even humans take pollen tablets as a rejuvenating energy source. Then hydrophilic. In hydrophilous flowers, means water pollinating. The pollen is ribbon-like with mucilaginous sheath, as you see in Zostera. Cryopreservation. Minus 196 degrees at liquid nitrogen, you're going to preserve the pollen. Next, male gametophyte, we already discussed. Asymmetric spindle formation during mitosis and pollen grain results in the formation of large vegetative and small generative cell. You could see this, children. This is the exine wall, intine wall, vacuole, nucleus. Spindle is formed on one side, dividing nucleus, 2 cells. This is generative, this is vegetative. At the Sood cell stage is liberated. Then you could see the pollen tube extending out from the vegetative. Then the generative cell divide, and you could see the 2 male gametes. Male gametes are non-motile, hence hyphenogamous type of gumi. Most of the cases, the pollen is released at 2 cell stage. Rarely in 30% 3 cell stage. And what is this transfer of pollen from anther to stigma called? It is said to be pollination. All right. And you know pretty well, the development of male gametophyte is partly in pollen sac, partly on stigma, isn't it? At 2 cell stage is getting liberated. After reaching the stigma, the pollen tube is formed, and the generative cell inside the pollen tube is going to divide and produce 2 male gametes. So part of the gametophytic development is also occurring on stigma. Autogamy. What is the meaning of autogamy? If the pollen of the same flower reach the stigma, that is said to be autogamy. Commelina, oxalis, and viola. I want to tell you, children, in these 3 plants, the flowers are underground, clistogamous. And the flowers are underground and they are closed. So naturally, they should be always bisexual. Autogamy occur. Is it a mechanism to ensure seed production? Yes. They don't want any variations. They want compulsory seeds. Clistogamy is a nice inbreeding device. Very important. And remember the examples. Commelina, oxalis, and viola. These are clistogamous and they are underground flowers. Then what is geitonogamy? The meaning of geitonogamy is the transfer of pollen from anther to stigma between 2 flowers on the same plant. So is it cross-pollination? Yes. But it is more or less the results of it is similar to self-pollination. Why? Because it is occurring within the same plant. Even though for namesake it is cross-pollination, but the results are similar to self-pollination, that is geitonogamy. Then what is xenogamy? The transfer of pollen from anther of one flower on one plant to anther of another, to the stigma of another flower of another plant. So pollination is involving 2 plants. Listen, pollination in one flower, autogamy. Pollination between 2 flowers on the same plant, geitonogamy. Pollination between two flowers present on two different plants, xenogamy. That point is important. Now, if I'm a plant with bisexual flower, then all three are possible. Autogamy is possible, geitonogamy is possible, xenogamy is possible. Isn't it? Imagine I have got male and female flowers, not bisexual, male and female flowers on me. Autogamy is not possible. Male flower, how can autogamy take place? So only geitonogamy and xenogamy is possible. If I have two types, such plants are called monoecious plants. Castor and maize, monoecious. Remember this word, monoecious. Male and female flowers present on the same plant. Then sometimes, entirely I'm male plant, entirely I'm a female plant. Because on me only male flowers. On me only female flowers, either male or female. Such plants are called dioecious. You have seen carica papaya plant, children. All the two plants are adjacent to one another. In one plant you see many fruits. In another plant there are no fruits at all. You think that the other plant is diseased. No, it is not diseased. It is male. All right. So it means that that's a very important point to remember. Plants with only one type of flower, xenogamy. That is, I repeat, plants with bisexual flower shows autogamy, geitonogamy and xenogamy. Plants with male and female flowers on it, that is monoecious plants, show geitonogamy and xenogamy. And the, and the plant which is dioecious shows only xenogamy. So shall I say that xenogamy is common in all three? Yes. Cleistogamy, pollination in a closed flower, we already discussed, it's an inbreeding device. Then what are these outbreeding devices? These are all outbreeding devices, children. Dicliny, dichogamy, hercogamy and self-sterility. What are outbreeding devices? If I'm a flower, I want always variations. All right, variations are possible when I get the pollen from other plant or the same species. So therefore, I tend to prevent or avoid self-pollination. Dicliny, if I'm unisexual, no chance for self-pollination. It's an outbreeding device. Dichogamy, male, I'm a bisexual flower. Stamens and carpels in me ripen at two different times. So naturally, there is no chance for self-pollination. Only cross-pollination can occur. Then hercogamy. I'm a bisexual flower. Stamens and carpels are at two different heights. So no chance for any self-pollination. All right, only cross-pollination occur. That is said to be hercogamy. This is very important. Self-sterility. I'm a plant, I have a flower. The pollen grains are viable. Pollen grains are not non-viable. They are viable pollen. But they germinate on the stigma of other flower only. They doesn't germinate on the stigma of the same flower. That is the meaning of what is called self-sterility. All right, so these are all outbreeding devices. Means it's a methodology to promote cross-pollination and at the same time prevent self-pollination such that more variations are possible. Then what is the meaning of right pollen? We already discussed. Pollen from same species is right pollen. Or self-compatible pollen is a right pollen. Then what is wrong pollen? Pollen from other species is wrong pollen. Self-incompatible pollen, I was talking about self-sterility, abetalon, passiflora. In all of them, the pollen will not germinate on the stigma of the same flower, but can germinate on the stigma of other flower. This is genetically controlled. That self-incompatible pollen is called wrong pollen. A chemical interactions occur between the stigma and the pollen. And after that, it is now up to the stigma to either allow the pollen to germinate or prevent the pollen from germination. This is a chemical interaction between the pollen and stigma secretion determined either to accept or reject the pollen. Pollen tube extends to the style and enter into the ovule. We already discussed the generative cell of pollen divide and produce two male gametes. They are non-motile, remember, and carried by pollen tube. This is called siphonogamy. What is the cell which produces the pollen tube? Siphonogamous cell, vegetative cell, because it produces a pollen tube. Then what is a spermatogenous cell of pollen grain? Spermatogenous cell of the pollen grain is generative cell, because it is the generative cell which is dividing and producing two male gametes. Two male gametes are produced from each pollen grain, yes, and 8 male gametes are produced from meiocyte. Naturally, One microspore mother cell produce 4 microspores. Each microspore produce 2 male gametes. 4 into 2, 8. All the changes, this is important, from pollen germination to the entry of pollen tube into the ovule is called pollen pistil interactions. Then I was talking about young anther and mature anther children. Look at the differences. This is young anther, where you could see the 4 theca very much intact. You could see the tapetum, you could see the middle layers, you could see the endothecium, and you could also see the epidermis. When you could see the 4 theca very clearly, it means that it is a young anther. But in a mature anther, the theca are vanished, means the endothecium is consumed. When the endothecium is consumed, when the middle layers are consumed, then both this put together becomes one chamber. Both this put together becomes one chamber. Then it becomes bilocular. That is a mature anther. I'm going to show you the diagram. And when we talk about the anther wall, children, I told you this is the anther. This is the anther wall. The outermost layer is epidermis. Inner to that, endothecium. Inner to that, middle layer. Inner to that, surrounding this microsporangium completely is tapetum. All right. So tapetum, that's a very important point. So epidermis, all these 3 layers, which 3 layers? Epidermis, endothecium, and middle layers. All 3 have a common function, that is protection. Apart from that, endothecium. I told you at a region called stomium, the epidermis break. At a region called stomium, epidermis break. This is the anther wall, children. This is the anther wall. You could see it, anther wall. Now this region, at this region, the epidermis break. Look here, epidermis break. This region, epidermis broke. And this region, epidermis break. Can you see the inner paper clearly? What is this inner paper? Endothecium. Now the endothecium is exposed to outside dry weather. Now what happens when they're exposed to outside dry weather? The endothelial cells lose water. They bend inwards. Don't you see a longitudinal slit on this side of anther lobe? One more anther lobe. So that region of epidermis, which is going to break and expose the endothecium to outside dry weather, is said to be stomia. How many stomia exist in a dithecous anther? On this side 1, on this side 1. So 2 stomia. Endothecia, I already told you, it is going to lose water and is liberating the pollen grains. Middle layers, helpful in protection. This is important. This is very important. Tapetum. It is the innermost layer surrounding the pollen sac completely. This, don't forget, very important. It is binucleate to multinucleate with loads of reserve food, helpful in nourishment of not only pollen mother cell but also pollen. Finished. Successfully, we have completed. Sporophyte means plant. Sporophyll means stamen. Sporangium means bag. Spore mother cell inside the bag. Reduction division spore, pollen grain. Microspore is nothing but the pollen grain. All right. Then gametophyte. 2-celled pollen grain is gametophyte. 3-celled pollen grain is also a gametophyte. Then last is gametes. You see, as you have seen, the generative cell dividing and producing 2 male gametes. This is looking simple. All right. Same way, we should talk about the megasporophyll. Isn't it? We have produced male gametes. Now it is time for us to produce the female gametes. Is it complicated? Why? Because the same table we follow. Sporophyte, microsporophyll, microsporangium, microspore mother cell, microspore, male gamete, male gametophyte. No. Sporophyte, sporophyll, sporangium, spore mother cell, spore, male gametophyte, male gametes, isn't it? Same way for the female reproductive system. Sporophyte, megasporophyll, megasporangium, megaspore mother cell, megaspore, female gametophyte, female gamete. So easy to remember. Now you are very good in that table. All right, how to remember the table? Leaves are seen on the plant. So plant, after that leaves, fill. Leaves will have bags, sporangium. Bags contain mother cells, spore mother cells. Reduction division to produce spore. Spore will give rise to gametophyte. This is one area you have to remember. Sporophyte, spore will give rise to gametophyte. And what is the meaning of gametophyte? Phyte ante plant. Plant producing gametes. So the job of gametophyte is to produce gametes. That's it. So let us now see. Then the next one is megasporophyll. Look here, children. This is the megasporophyll. It contains ovary, style and stigma. Fill. Fill means leaf. Look here, leaf. This in gymnosperms, this leaf is open. Open carpels. What about in angiosperms? Look on the midrib. In first stamen, I've removed this, I've broken this, tear this book into two parts. Each half I have folded like this. This is one lobe, and one more part I have folded one lobe. So two lobes, dithecus I told you, 4 chambers. Then what about the megasporophyll? Again, I take a leaf. Look here, this is a leaf. Now I'm folding like this. That's it. Okay, no tearing the book. Only one. This is the ovary. On the top, style. On the top, stigma. So ovary, style and stigma. This is megasporophyll. This carpel is open in gymnosperms. Means there is no ovary. No ovary, no fruit. That is the reason gymnosperms doesn't produce any fruits. Alright. This is the ovary, style and stigma. This is said to be megasporophyll. We'll spend some time here, children. There are 5 carpels. And you know what are how 5 carpels are united? We already discussed in axial placentation. This is one carpel which is folded like this. One carpel. Okay. This is the dorsal suture on the line which this is the ventral suture. If I put it here, dorsal ventral. One more carpel, dorsal ventral, one more. So like this, all the 5 carpels united to form one ovary. Alright. So this is the ventral suture. These are the ventral sutures. And on the ventral sutures, ovules are attached. So is it true that in hibiscus, all the 5 carpels are fused? Yes. Styles are also fused, but stigmas are free. Very important. Look at next example, papaver. Papaver is not stylish. All right, I mean, there is no style here, okay? It contains the ovary and on it there is a stigma. There is no style. This is an important. This is the ovary and this is the stigma. You don't see style here. That is papaver. All right. Then this is mycelia. This is thalamus. You could see 1, 2, 3, 4, 5, 6, 7, 8. So all the carpels are free. What about hibiscus? You could see 5 carpels, but all of them are fused to form one ovary. Here all the carpels are free. Then every carpel will have one ovary? Yes. Then each ovary become one fruit? Yes. There are many fruits in one flower? Yes. Such type of fruit, aggregate fruit. Custard apple, strawberry, mycelia, lotus, rose. They are all examples of aggregate fruits. Alright. Chalo, if what I have written, now we completed megasporophyll, right? And this is later we'll come across, okay? First we'll get rid of megasporophyll. If all the carpels are free, then it is said to be apocarpus. If all the carpels are fused, then it is said to be syncarpus. Hibiscus, 5 free stigmas. Papaver, style is absent. Mycelia, many free carpels. That's a very easy one to remember. Why this white page? I want to show you how the megasporophyll will produce megasporangium, megaspore mother cell, megaspore and female gametophyte in 2 minutes. Okay, chalo, let us see. This is megasporophyll. We completed this. Then what do you see after sporophyll? You should remember, by this time you should know the table. After sporophyll, sporangium. Inside this ovary, there is a cavity called ovarian cavity or locule. There you see sporangium. There is a cushion tissue called placenta, from where the sporangium, look here, this is the bag. This is the bag which is developing from the cushion tissue called placenta. And this bag is said to be sporangium. So let me draw the diagram of sporangium. This is a bag. And this bag is covered by an integument. You could see. Somewhere you have seen this structure. Yes, children, you guessed correctly. This is not only megasporangium but also ovule. What is this stalk called? Funicle. What is this scar called? Hilum. What is this entire part called? Body of the ovule. What is this called? Integument. What is this called? Micropyle. Isn't it? Micropylar end is said to be anterior end, chalazal end. This is the chalaza. The chalazal end is said to be the posterior end. This ovule is a straight ovule, orthotropous. In gymnosperms mostly and some primitive angiospermic families, you see this type of ovule called orthotropous. I'll come back to the most common ovule, anatropous ovule, after a minute. This is it. Don't forget, we are going through the table. After sporophyll, sporangium, this is the bag. It is covered by either one or two integuments. If it is one integument, unitegmic. If it is two integuments, bitegmic. Outer integument become outer seed coat called testa. Inner integument become inner seed coat called tegument after fertilization when this ovule becomes seed. All right. That's all. That's about megasporophyll and megasporangium. What is the next one after megasporangium? Megaspore mother cell. You're following me. You're following me properly now. You're concentrating what I'm talking now. All right. Now this is the nucellus. Yes. Nucellus. This is the nutritive tissue called nucellus. One of the nucellar cell towards the micropylar end act as archisporial cell, not archegonium. Archegonium is a female sex organ seen in bryophytes, pteridophytes and gymnosperms. That is archegonium. This is archisporium. Now, is it a diploid cell? Yes. This cell undergo mitotic division and produce two cells, parietal cell and sporogenous cell. So the sporogenous cell will act as megaspore mother cell. What I have written here is archisporial cell initially. Now I can even call this as megaspore mother cell because archisporial cell undergo mitotic division and produce two cells. One of the cell is megaspore mother cell. Is it true, children, to say that megaspore mother cell is formed towards the micropylar end of the ovule? Yes. All right. Now go back to the table. Sporophyll, megasporophyll, sporangium, megasporangium, and this is megaspore mother cell. What do you see next? Megaspore mother cell undergo reduction division and give rise to megaspores, isn't it? Now look here. It undergo reduction division and give rise to megaspores. Because there is only one megaspore mother cell, there will be only 4 megaspores inside one ovule. 4 megaspores. What next? Out of 4 megaspores, 3 megaspores present towards the micropylar end degenerate. Nice. Then 1 megaspore towards the chalazal end of the ovule will be functional. Careful. Megaspore mother cell arise from a cell called archisporium present towards the micropylar end, megaspore mother cell. Whereas functional megaspore is present towards the chalazal end or posterior end of the body of the ovule, not towards the anterior end. That's an important point. What do you see? Sporophyll is over, sporangium is over, spore mother cell is over, spore is over. What next? After spore, the next is gametophyte. Yes or no? Gametophyte. Now what will happen to this spore? Please concentrate. This, I'm drawing this outside. Don't think that it is thrown out. All right, only for convenience. This is the megaspore. What will happen? This undergo free nuclear divisions and the 2 nuclei migrate towards the 2 poles. So now free nuclear division is nothing but mitosis. Karyokinesis took place but not cytokinesis. So now in place of 2 cells, say 2 nuclei. one nuclei migrate towards one pole, other will migrate to other pole. This is after one generation of mitosis. One generation of mitosis. Then what happens next? Then you see this nucleus divide into two. All right. And this nucleus will divide into two. All right, let me show you like this. So one nucleus, then you'll be seeing two nuclei. Two nuclei have become four nuclei. Then what happens next? Each nucleus divide further. So naturally, four nuclei here and four nuclei here. All right, then what will happen next? Listen carefully. One nucleus from both the poles migrate towards the center. One from here, one from here. Therefore, rightly called polar nuclei. Now what will happen? Now the wall formation will occur around this nuclei. So how many nuclei present here? 1, 2 and 3. How many nuclei present here? 1, 2, 3. First, it is one nucleus. Next, 1 plus 1 nuclei. Next, 2 plus 2. Next, 4 plus 4. Next, 3 plus 2 plus 3. Now the wall formation occur. Look here. This is the wall formed. This is the wall formed and wall. Now you can see the wall is formed around 3 nuclei there. This is the wall formed and formed. Wall is formed around the 3 nuclei here. All right. Let us count how many cells are there. 1, 2, 3, 4, 5, 6 and 7. How many nuclei are there? 1, 2, 3, 4, 5, 6, 7, 8. All this is developing from megaspore. What spore should give rise to? Gametophyte. Can I call this as female gametophyte or embryo sac? Yes. It contains 7 cells and 8 nuclei. Then 7-celled, 8-nucleated megaspore is said to be female gametophyte or embryo sac. All right. How is this, sir? How many mitotic divisions? The next question you're going to ask me is, first let me show you here. This is sporophyll, this is sporangium, this is spore mother cell, this is spore, this is the functional spore, this is female gametophyte, this is the egg apparatus, and this is the female gamete. This is the female gamete. Yes or no? Sporophyll, sporangium, spore mother cell, spore, functional spore, female gametophyte, female gamete. So easy to remember. All right. Then if you look at how many mitotic divisions are needed to produce a fully developed female gametophyte, let us count here, children. This is one nucleus. All right. It has divided into 2 first. Yes. Then again it is divided into 2. Yes. Again this is divided into 2. Yes. Again this is divided into 2. 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 Yes or no? Now count how many nuclei? 4, 8. Let us see how many mitotic divisions. Let us count. 1, 2, 3, 4, 5, 6, 7. Good point. There are 7 mitotic divisions needed in the megaspore nucleus to develop into a fully developed female gametophyte. But if they ask you how many generations of mitosis, this is 1 generation, this is 2 generations, and this is 3 generations. Means 1 becoming 2 becoming 4 becoming 8. If they ask you generation, this is 1 generation, 2 generation, 3. So the number of generations of mitosis are 3. Whereas the number of mitotic divisions, if they ask you, the answer is 7. This is very easy to remember. Now, this is sporophyll, I told you. Let me also show you the microsporophyll here. Then both of them put together, we'll see. This is microsporophyll, yes. And this is megasporophyll. This is nice, easy to remember. Then after sporophyll, sporangium. This is microsporangium. And this is megasporangium. So easy to remember. Then these are microspore mother cells. There are many. inside each sporangium. This is interesting. Whereas here, there is only one megaspore mother cell inside the megasporangium. Be careful. Then what the next one? After spore mother cells, the next is spores. This is a microspore, pollen grain. And this is a megaspore, functional megaspore. What next? Then what next? Then the spore should give rise to dash. The spore should give rise to gametophyte. Yes or no? Then this is going to develop first into 2 cells. Then a 2-cell stage is liberated, pollen tube. Then it divides, the generative cell produce 2. This is fully developed male gametophyte. Coming to the female, this is going to undergo 7 mitotic divisions and produce an egg cell. This is turning out to be easy. Sporophyll, sporangium, spore mother cell, spore, microspore, megaspore. Fully developed male gametophyte, fully developed female gametophyte, male gametes and female gametes. Let us see what I have written with regard to the megasporogenesis, children. Please concentrate here. Now it's very easy now. We'll be going through very, very fast. All right. With regard to the megasporangium. We stopped at megasporophyll. Now we'll go to megasporangium. Chalo. Now where it is seen? Megasporangium is seen in pteridophytes, gymnosperms, angiosperms. Interesting. Even in pteridophytes. Heterosporous pteridophytes like Salaginella and Salvinia. Megasporangium with an integument, that is ovules. Integument that is seen in gymnosperms and angiosperms. Only these can produce seeds. Salaginella is deprived of seeds. All right, as it lacks integument around megasporangium, though it has got many precursor characters. Bad luck. It shows many precursors, including the female gametophyte is retained on the parent sporophyte for many generations. In spite of showing all the precursor characters, it lacks seeds because it doesn't have integument. The most common type of ovule is anatropous. Look at this ovule, children. This is the funicle. It is inverted ovule. This is the embryo sac. And don't forget, the egg apparatus will be always towards the micropellar end. Remember. And these are the antipodals. And this is the central cell. This fusion area is said to be raphe. The fusion area of funicle with the integument is said to be raphe. This is the inverted ovule, anatropous. Straight ovule is orthotropous. The stack funicle. Now we can just run through it. The basal part of the body of the ovule, chalaza. The fusion area of funicle with the body of the ovule is hilum. Ovules possess either 1 or 2 integuments. The micropyle is a small opening, you know that. Micropylar end is anterior and the chalazal end is posterior. Inside the ovary, there are cavities called locules or ovarian cavities. Ovules arise from a special tissue called placenta. One of the nucellar cell towards micropyle differentiate into megaspore mother cell. That nucellar cell is called archisporium. The number of megaspore mother cell inside an ovule is always 1. Number of functional megaspores inside one ovule is always 1. The functional megaspore mother cell, we already discussed this, archisporia. The functional megaspore is the one which is present toward the chalazal end. You know that. All right. Megaspore mother cell after meiosis produce 2 cells. Look here. These are the 2 cells. After meiosis 1, 2 cells. And after meiosis 2, this megaspore mother cell produce 4 megaspores. 3 megaspores disintegrate. The chalazal megaspore will be functional. Megaspore develop leading to the formation of female gametophyte or embryo sac. Megaspore undergo free nuclear divisions. Oh, very important. I have highlighted all these points properly, children, such that it's very easy for you to run through. All right. Mitotic division 7. Generations of mitosis 3. Mitotic divisions at each pole. At each pole, 1 nucleus is becoming 2, 2 has becoming 4. At each pole, the number of mitotic divisions will be 3. This is 1 mitotic division, yes or no? And this again dividing into 2, this again. So 3 mitotic divisions in total at each pole. Mitotic divisions at both the poles 6. Total number of nuclei 8, cells 7. Valve formation occur around 6, we already discussed. And you could see this filiform apparatus. These are the 3 cells, egg apparatus. You could see the egg cell is bigger and the synergid cells are relatively smaller. Filiform apparatus secrete chemicals and guide the pollen tube towards the egg cell. These are called antipodals. Easy way to remember antipodals. SSS. Smallest cells of embryo sac, short-lived cells of embryo sac, somatic or vegetative cells, because they doesn't have any role to play. Those are called antipodals. And these 3 cells put together is said to be egg apparatus. Don't get confused between embryo sac and egg apparatus. You have to be very careful. Embryo sac is 7-celled, 8-nucleated. It contains 8 nuclei. So therefore 8 genomes. It is haploid. Don't say 8N. All right. It is haploid. It is polygonum type. It is also called monosporic. That 7-cell, 8-nucleated is said to be monosporic or polygonum. And only one embryo sac will be there in one ovule. Only one pollen grain is needed to fertilize one ovule. Why? One sperm fused with egg, other sperm fused with these 2 polar nuclei. 2 sperms will be there inside a pollen grain. So one pollen grain is enough. The 2 male gametes are needed to fertilize one embryo sac. Why? Because one male gamete to fuse with egg, other with 2 polar nuclei. The number of female gametes will be 1. Antipodals, already I told you, present toward the chalazal end is very important. Smallest cells, short-lived cells, somatic or vegetative cells. They may degenerate either before or after fertilization. Egg apparatus, mind you, egg apparatus. 3 cells towards the micropylar end or anterior end. It contains 2 synergids and 1 egg cell. Synergids are the smallest cells of egg apparatus. Be careful. Smaller cell of embryo sac means antipodals. These are the very important. Filiform apparatus, this is only 1 in number, be careful. And it is seen in synergids. Filiform apparatus secrete chemicals and guide the pollen tube towards the egg cell. Alright, this is said to be chemotropism, outside chance. Egg cell is the largest cell of egg apparatus, okay. Then this entire thing is called central cell. It is the largest cell of embryo sac. Initially it is binucleate because it has got 2 polar nuclei. Later the 2 polar nuclei fuse to form secondary nucleus. It has a large vacuole and peripheral cytoplasm. Increasing order in the size in the cells of embryo sac, smallest antipodals, next synergid, next egg. This is an important question. Then with the last 2 parts, children, pollinating agents we'll talk about here. Nothing special for me to talk. First is anemophily, wind pollination, most common. Abiotic. Within abiotic, it is the most common. And in all gymnosperms, it is wind pollination. Grass family of monocots, mostly it is wind pollination or anemophily. Flowers are generally small. Perianth is reduced or absent. Lodicules in Poaceae members, lodicules we call. Stamens just come out, exerted. Pollen is produced in large quantities, plenty. And it is dry and light such that it can be dispersed. Why in large quantities? Because the dispersal of pollen is a chance factor. Okay. So if it has to fructify, the pollen should be produced in large quantities. Ovules inside the ovary should be only 1. Why? Because even if a single pollen reaches the stigma, the act of fertilization can occur. So therefore, having minimum number of ovules inside the ovary is always a good idea. Fixation of stamen is versatile. The anthers can rotate like this such that it can, this point is important. Tassel and ear, look here. This in a maize, this is said to be tassel. Tassel is a male organ, remember this. The job of tassel is to produce pollen grain. And this is ear. And you know it's a monoecious plant. You know that, male and female flowers on the same plant, ear. And these are the silky structures. These are styles and stigmas of the ear. Ear represent female inflorescence, okay? And these are the silky styles and stigmas which catch the pollen. So therefore, next time no confusion. Tassel is a male organ, ear is a female organ. In the new NCERT book, they replace tassels with ear because ear contain many flowers. These flowers contain silky styles and stigmas which catch the pollen. Then we go for hydrophily, water pollination. It is less common abiotic agent. Very important. Only submerged hydrophyte shows water pollination. Free floating and emergent hydrophytes mostly show insect or wind pollination. Very important. Flowers, sometimes they may get detached as you see in Vallisneria. Pollen is surrounded by mucilage in a sheath for protection. Plenty and ribbon-like pollen is seen in Zostera, which is seagrass, both in abiotic, wind and water. Pollen grains are produced in large quantities because transfer of pollen is a chance factor. And example for hydrophily, Vallisneria, Zostera, Hydrilla, Ceratophyllum. Look at Vallisneria, children. I'm going to show you here. This is the male plant and female plant, so it is dioecious. You could see the male flowers getting detached and reaching the surface of water. The female flower uncoil, the pedicel of the female flower uncoil and bring the female flower onto the top of the surface of water. Pollination occur on the surface, hence epihydrophilic. And what are the points about Vallisneria? Epihydrophily, dioecious plant, it is a freshwater plant. Male flowers get detached, female flowers reach the surface with the help of long pedicel, pollination on the surface, hence it is rightly called epihydrophily, Vallisneria. Zostera, it is a sea grass, don't say seaweed, brown alga is seaweed. It is marine, name itself, sea, marine, ribbon-like pollen, and the pollination occur inside water, hydrophily. These 2 points are important. In majority of hydrophytes, wind and insect pollination is very common, as you see in water lily and water hyacinth. Whereas in alga, bryophytes and pteridophytes, water is needed for fertilization, not pollination. Very important. Then the entomophily, mostly with the help of bees. All right. Most common biotic agent. This is mostly seen in dicots. Flowers are large and attractive with scent glands. Why scent glands? If the flowers are nocturnal, the insects can easily what is called move towards the flower if they have got scent glands like jasmine, cestrum. All right. Then perianth, it is colored to attract the insects. Stamens, less pollen in comparison to abiotic, nothing wrong in it. Pollen is sticky, very important, and edible. Edible to whom? Edible to insects as well. All right, sticky and edible. Ovules inside the ovary, it could be one to many. These are important, floral rewards. So to attract the insects, the flower is going to announce some rewards. And what are those rewards? Nectar glands, so the insects can drink. Edible pollen, the insects can eat. Safe passage for insects to lay eggs, means insects can sleep. Drinking, eating, sleeping, all three the flower is going to provide. Nice. All right. And the insects surely will come. Amorphophallus and carrion flies is one example for safe passage for laying eggs. I'm giving an example. Yucca and moth. Moth is going to search for yucca and move towards it. Ficus and wasp. These are all the examples which you have to remember, where the flowers are going to provide safe passage for the insects, such that the insects can come in large numbers and pollinate them. Male gametes we produce, female gametes we produce. It's only a formality that we have to see that the male gametes fuse with the female gamete. And all of you know that this is the pollen tube. It is extending. It has got two male gametes. This is the ovule and this is the embryo sac. And this is the egg cell and this two polar nuclei. The pollen tube enter into the synergid. Synergid. Why? Because synergids secrete chemicals. The male gametes get liberated. One of them fuse with egg and the other fuse with these two polar nuclei. The male gamete fusing with female gamete is said to be syngamy. First I talk about syngamy. Number of male gametes, 1, correct. Number of female gametes, yes. Number of paternal, male means paternal, 1. Number of maternal genomes, 1. The product is zygote. The ploidy of zygote is diploid. Observe from alga to angiosperms. Syngamy, fertilization. Then what is triple fusion? One of the sperm fused with these two polar nuclei. 3 nuclei are fusing. That is said to be triple fusion. Let us talk about triple fusion. Number of male gametes, 1, correct. Number of female gametes, none, correct. There are no female gametes. Number of paternal genomes means male, 1. Number of maternal genomes means female. All right, 2. Did all of you understood this? Then product, primary endosperm cell, and the ploidy is triploid. And it is observed only in angiosperms. And what are these two put together called? Syngamy and triple fusion. Double fertilization. Very simple. The number of male gametes involved will be 2. The number of female gametes involved will be 1. Number of paternal genomes 2, because there are 2 male gametes. Number of maternal genomes 3. 1 egg cell, 2 polar nuclei 3. Product zygote and primary endosperm nucleus. Zygote is diploid, endosperm is triploid. It is seen only in angiosperms. The number of nuclei involved in double fertilization, the answer is 5. Remember that. Then artificial hybridization to produce hybrid seeds, what we do. In a crop improvement program, the desired pollen grains of one desirable plant, you artificially pollinate on the stigma of another. That is said to be artificial hybridization. It includes emasculation, bagging, artificial pollination, and the production of hybrid. Why you have to remove the stamen? Naturally, children, I want the pollen grains of this flower to reach the stigma. This flower also have stamen. No, I have to remove it. That is called emasculation. So this is the female parent. Why you say female parent? Because the characters of egg you want to exploit. Not that it is unisexual female. It is bisexual only. The characters of egg you want to exploit. Therefore, it's a female parent. So from the female parent, you remove the stamen so as to prevent any self-pollination. The same female parent flower, you're going to cover with a butter paper bag such that unwanted cross-pollen doesn't occur. Then I open the bag, take the pollen grain by using a brush, and I'm going to sprinkle on the stigma of this female and cover the bag again, such that the characters of desirable nature of both the parents will come together, resulting in the formation of an hybrid. So emasculation is the removal of anther from a bisexual flower bud of a female parent. All right. It is not needed if the female parent is unisexual or if it is male sterile. By biotechnology, you can produce male sterile flowers in mustard. It prevents self-pollination. Emasculation prevents self-pollination. Bagging. Emasculated flowers are covered with a suitable bag or to prevent unwanted cross-pollination. You can't avoid this. Bagging is a compulsory process. Artificial pollination, you take the pollen of that and drop it on the stigma. That is very important. The final products of sexual reproduction are fruits and seeds. Now, what a typical seed possesses? A seed will have endosperm, embryo along with seed. E for endosperm, E for embryo. All right, apart from that, seed coat. Now, coming to the endosperm. Come on, you know it. Ploidy is triploid. The product is triple fusion. We had last 3-4 minutes, children. Just concentrate here. Develop before embryogenesis. Why? Because only when endosperm is produced in large quantities, it can provide nourishment to the developing embryo. Short-lived. Endosperm is short-lived in dicots. That is the reason they are called non-endospermic. Some of you are believing that non-endospermic means endosperm is not at all formed. Wrong. Endosperm is formed but short-lived. Dicot with persistent endosperm, very important. Castor and solanaceae. Endosperm is long-lived in monocots, hence albuminous seeds or endospermic seeds. Monocot with short-lived endosperm is orchids. This is an exception. Normally, monocot should have long-lived endosperm, but it is short-lived in orchids. Endosperm storing oil, castor oil, it's a dicot. Coconut, it's a monocot, isn't it? That white-colored kernel is nothing but what is called the endosperm, from where you get oil. So therefore, a monocot with oily endosperm, coconut. Dicot with oily endosperm, castor. Endosperm storing proteins is aileron layer. You know, children, rice. This is the scutellum. This is the radical plumule. You know, this is the layer which is said to be aileron layer. And this is the starch part of endosperm. All right, and this is the protein part of endosperm. That is said to be aileron layer. Major part of endosperm in cereal store starch. The outermost layer contain proteins. Gibberellins are synthesized in embryo and they move to the aileron layer. Within the aileron layer, they synthesize alpha amylase, we discussed, that migrate to the starch part and convert them into maltose. Embryo. After endosperm formation, embryo occur. Look here, children, first zygote. It develops into proembryo. Proembryo means that stage which has not transformed into any particular shape. All right. Then globular embryo. This is globular embryo. Then heart-shaped embryo. When you could see the Cotyledon primordia, it becomes heart-shaped embryo. Then you see a mature embryo. This is the mature embryo. So what is the meaning of proembryo I have written? Zygote undergo its first mitotic division. It produces 2 cells, children, like this. All right. Then the basal cell becomes suspensor. One of the cell becomes suspensor. Look here, this cell. And this cell is going to undergo many mitotic divisions like this. All right. And this is said to be what is called the development of embryo. The basal cell becomes a suspensor. It pushes the developing embryo deep into the endosperm. A few successive divisions in the terminal cell produce a group of undifferentiated cells. This young multicellular structure without any shape is said to be proembryo. What happens next? Proembryo later becomes heart-shaped embryo. Sorry, globular embryo. It becomes round. Then it becomes heart-shaped embryo once the cotyledonary primordia develop. Then it becomes a mature embryo. Now here I have shown you the difference between the embryo of dicots. The embryo of dicots have two cotyledons. Look here, simple diagram I'm drawing for convenience. All right, look here. These are the two cotyledons. This is an embryonal axis. This is the radicle. This is the plumule. This is hypocotyle. This is epicotyle, because this is present above the node. Actually, it is not like this. It's somewhere like this. But for convenience, I have shown like this. This is a dicot embryo. It is developing from zygote. Mature embryo. Two cotyledons, embryonal axis, radicle, hypocotyle, cotyledonary node, epicotyle, and plumule. If it is a monocot, children, there is only one cotyledon. There is a reduced cotyledon here, which is said to be epiblast. This is said to be scutellum. Coleoptile, coleorhiza are the protective coverings which are surrounding it, and this large shield-like cotyledon is called scutellum. First, we look at dicot. It has got two cotyledons. All right. Heart-shaped during development because of leaf primordia. Cotyledons store plenty of food. Plumule production, radical production is absent. All right. What about monocot? One cotyledon, the other cotyledon is reduced, called scutellum. And the cotyledon which is well developed, shield-like, is said to be scutellum. Of course, in grass family, not in all. All right, scutellum absorbs food from endosperm. Plumule is enclosed by coleoptile, vent. We discussed vent experiment. Radical by coleorhiza. So this is globular embryo, caudate embryo, and mature embryo. Cotyledon and mature embryo will have, this cotyledons, plumule, radical, hypocotyle, epicotyle. I already told you. Epiblast I talked about. Scutellum also I talked about. We have the last 3 minutes, children, where we talk about the fruit and seed. Fruit. Ovary after fertilization is fruit. And where do you see fruit? It is seen only in angiosperms. All right. Then apple. Some of you got a doubt. What I eat an apple? Apple a day keeps a doctor away. All right, which apple? False fruit a day, not the true fruit. True fruit, if you eat, you have to go to doctor every day. All right, so what is the false fruit? How it is formed? Look here. This is the thalamus, no? Just simple example I'm talking about. This is the thalamus. But in apple, the thalamus is going to become, look here, deep cup-shaped. Look here. The thalamus have become cup-shaped. So this is the cup-shaped thalamus which you eat. Then you may ask me, ovary? Ovary will be there. And this is the style ovary. The ovary will become fruit. So if you take a section of this, if you take a transverse section of it, this will be like this. This is the ovary. 1, 2, 3, 4, 5. 5 carpels, just like hibiscus. 5 carpels, just like hibiscus, united to form that black colored, brown colored structure inside the apple is the fruit, true fruit called pome. All right, if you take a section of that, you see 5 carpels united with the axile placentation. Strawberry, again, the thalamus is edible in strawberry. And the true fruits are achenes. Those points are important. False fruit can develop from any other part of the flower other than ovary. False fruit in apple is thalamus. The true fruit is pome. False fruit in strawberry is thalamus. The true fruit, the juicy part in strawberry is thalamus. And the true fruit, you're going to see small dots like this. So these are all the true fruits, which we call them as achenes. All right. Fleshy fruit example, berry, pepper, pome. Berry will have pulp. Pome is the fruit, true fruit of apple. Pepper is cucumber. Drupe is mango. Dry fruit, remember, legume, bean, mustard, siliqua, cypsela, and caryopsis. This you have to remember. Coming to seed. All right. Seed is a product of fertilization. And seeds are seen in gymnosperms and angiosperms, hence spermatophytes. Seeds contain seed coat, embryo, and endosperm. One seed per fruit is very common in grass family, Not only grass, mango, even coconut. Many seeds, papaya, watermelon, orchids, striga and orabankia. These two are important. Remember that. Seed dormancy, temporary suspension of growth due to internal factors like hard seed coats, immature embryos is dormancy. And if it is through external factors like water, oxygen deficiency, you call it as quiescence. All right. Then next, children, what are the advantages of seed? We'll be talking about. What are the utility of seed? All right. First is it protects the embryo. All right. The embryo is enclosed within a tough seed coat. And one more, it has plenty of food either in its endosperm or in cotyledons. When the conditions are not favorable, it has a license to stay dormant. It can sleep when the conditions are not favorable. And it can be dispersed to far off places. You can just throw them or you can carry them to far off places. And it is able to survive the adverse conditions, just like underground stems. Perennation. All right. They form the basis for agriculture because they're important for propagation, storage, transport. And this is a very important point. It is dependable and trustworthy process. I can assure that an angiospermic plant always produces seed, even in the absence of water. Why? Because fertilization and pollination, the true prerequisites for the formation of seed, doesn't need any water. So the seed production is more dependable. Dependable means don't think parasite dependable. No, dependable means trustworthy. All right, then we talk about apomixis. Now what is apomixis? Just last 2 minutes, children. Sporophyte, yes, you see megasporophyll. Sporophyte produce megasporophyll. Let me write here. All right, this is the spore. Just listen for a minute. Sporophyte, yes. Then it produces dash. Megasporophyll, yes. Now listen here, children. The last part always, you tend to go in a hurry and make mistakes. Then it produces megasporangium. Yes, we already discussed this. Ovule is megasporangium. Now you are very good. Megaspore mother cell, one of the archisporial cell. Then it undergo reduction division and produce megaspores. Chalazal megaspore is functional. Megaspore develop into female gametophyte. Embryo sac, it is 7-celled, 8-nucleated, monosporic, polygonum type. All right. Then the female gametophyte will produce egg apparatus, and egg apparatus have female gamete. And this is going to fuse with male gamete, yes or no, to form a diploid zygote, and zygote develop into embryo. All of you know that. And this embryo will develop into root, stem, leaf and flower. That is said to be sporophyte. Let me show you like this. So now look here. Sporophyte, megasporophyll, megasporangium, megaspore mother cell, megaspore, female gametophyte, female gamete. It fused with male gamete to form embryo. Embryo form this. 1 minute, children, just 1 minute. All right. Do you see reduction division here? Do you see reduction division here? Where? Where? All right. This is it. Reduction division. Yes. Do you see fertilization here? Syngamy. Fertilization here. Yes. All right. When you see reduction division and fertilization, you call this entire thing by one name, amphimixis. Amphimixis. This is easy to remember. Now what I do, listen, this is very common. That's what we have discussed for the last 1 hour, 10 minutes. Little high, but totally we discussed. Okay. Now, now what I do, listen here, what I do now. I don't want any reduction division. I don't like reduction. Nobody likes reduction, isn't it? No reduction division. All right. I don't want this fertilization also to take place. All right. Then let us see what is the ploidy of all. Okay. Sporophyte will be generally diploid. Megasporophyll will be diploid. Megasporangium will be diploid. Megaspore mother cell will be diploid. When reduction division in amphimixis, megaspore will be haploid, Female gametophyte will be haploid. Female gamete will be haploid. Male gamete will be haploid, fused to form zygote, embryo, diploid. So when normal amphimixes occur, now there is no reduction division. Then interestingly, megaspore will be diploid. Female gametophyte will be diploid. Female gamete will be diploid. Are you listening? I told there is no fertilization, so no male gamete. So the embryo will be diploid. Don't say tetraploid, no male gametes, no. And this embryo develop into sporophyte diploid. You may ask me, tell me, is it sexual or asexual? I get the feeling that this is asexual. Why? Because there is no meiosis and there is no reduction division. And the progeny formed are exact replica of the parent. Correct. But gametes are formed? No. Gametes are formed. And where do you see gamete formation? In sexual reproduction. Then no confusion. Gametes are formed, but there is no fusion of gametes. There is no zygote. There is no fertilization. There is no reduction division. Therefore, it is definitely asexual reproduction, which mimics, means which imitates sexual reproduction because gametes are formed. What is the advantage of apomixis? No variations. Children, if I am getting a plant with all the desirable characters, imagine if this is a plant, this is A, all right, B, this is A, all right, then A, B, and this is a plant which is desirable. It has got all the dominant alleles, all right. Now, I want the same plant, same replica. In normal hybrid industry, it is not possible because once I sow the plant, maybe all the 3 desirable characters I can see, but there is no guarantee that they can be seen in the next generation because random cross-pollination can occur. So, if I want the same desirable genotype existential for many generations, then I should convert this plant into apomictic plant. Yes or no? Genetic control. Then I could see this, because there is no reduction division, there is no meiosis. I can guarantee that the same genotype exists. This is an advantage of apomixis, especially in hybrid seed industry, because hybrid seeds cannot be used again and again, all right, because of segregation of characters. But here there is no segregation of characters. Therefore, apomictic seeds are very important. Last point, this is a season of mango. All right, so I'll tell you one point about mango and we'll finish off things. Have you ever seen a mango fruit? Oh, what is this? Ridiculous. We have eaten many times. Next time, children, after eating mango fruit, all right, do this. Right, keep the seed. Seeds you don't eat, no? No. All right, keep the seed aside. Make it dry. Keep it dry. For a few days after that, it dry. Take a section of that seed. Take a section of that seed. Then you're going to see many tiny white colored structures, small globular structures inside. Seed should have embryo. It can't be endosperm. Definitely it should be embryo. Yes, but there are many embryos. You may ask me, why many embryos? Look here. One of them developed by amphimixis, means double fertilization. One of the embryo there is amphimictic embryo, sexual embryo. And the remaining embryos develop by the encroachment of nucellus into embryo sac. Can I call them as apomictic embryos? If nucellus is developing into embryo, there is no meiosis and reduction division. I can definitely call it as apomictic embryos. So, next time when you see mango seed and also citrus seed, take a section of them. You could see many embryos, out of which one is amphimictic embryo, means sexual embryo, remaining all are apomictic embryos. This is a wonderful example for polyembryony, because many embryos are there inside the seed. Finished. We have completed this chapter called sexual reproduction in a very nice manner, though little time taking, no problem. Right, thank you very much, and we'll be discussing one more topic tomorrow. Genetics we are going to start. That is very important. Please listen to that. Thank you.